Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th biology students, and this is for 18th November 2020. We are into the revision mode as you all know it, and we are revising all important chapter number 11, Biotechnology, Principles and Processes. In our last session, we were focusing upon processes of rDNA technology. After having discussed about the tools required for rDNA technology, now we are into the processes. As part of the processes, we reflected upon the 3 steps that we discussed yesterday. First step, isolation of genetic material, that is DNA. How to isolate DNA. Second, cutting and ligation of DNA at specific locations. Second step. And the third step that we discussed is amplification of gene of interest using a method called PCR, polymerase chain reaction. And in the PCR, we talked about the 3 steps that are involved. That is, the polymerase chain reaction, each cycle has 3 steps. Step number 1, denaturation. In this, the target DNA is heated to a high temperature, usually 94 to 98 degrees centigrade, resulting in the separation of 2 strands. Each single strand of the target DNA then acts as a template for DNA synthesis. This is denaturation. Obviously, when we heat the DNA at 94 to 98 degrees centigrade, naturally it gets denatured. Then second step that we talked is primer annealing. Annealing means joining. The 2 oligonucleotide primers anneal means join together, hybridize to each of the single strand template DNA. Since the sequence of the primers is complementary to the 3' ends of the template DNA. As you are seeing, this is the complementary one, 3 to 5', this one, whereas this one 5' to 3', and this is 3' to 5', and the complementary is 5' to 3'. And that is what we say. This step is carried out at low temperatures, usually 40 to 60 degrees. Look at that, 40 to 60 degrees centigrade. This annealing is done depending on the length and sequence of the primers, of course. And the third one that we talk about is extension of primers, that is polymerization. Other name for this is polymerization. Tuck DNA, this is important. Tuck, what is tuck? A bacterium named Thermus, T for thermus, and AQ aquaticus. So TAQ, tuck polymer. Tuck DNA polymerase from the thermophilic bacterium Thermus aquaticus synthesizes the DNA region between the 2 primers, as you are seeing here. You can see here, it's synthesizing it. And using DNTP. What is this DNTP? Deoxynucleoside triphosphates. And of course, Mg2 plus is a must. Magnesium 2 plus is a must, Mg2 plus. And it means the primers are extended towards each other so that the DNA segment lying between the 2 primers is copied. As you are seeing here, this is getting extended and amplified ones have been formed. And this occurs at an optimum temperature of 72 degrees. Polymerization or extension of the primer happens at 72 degrees centigrade. And we move ahead with the step number 4, insertion of recombinant DNA into host cell. How to put this into the host cell. The vector DNA or plasmid DNA and foreign DNA carrying gene of interest are cut by the same restriction endonuclease to produce complementary sticky ends. With the help of DNA ligase enzyme, the complementary sticky ends of 2 DNAs are joined, and this process, of course, annealing, to produce rDNA. Eukaryotic genes do not function properly when cloned into bacterial cell because of introns. In such cases, DNA is made from mRNA by reverse transcription or synthesized artificially. This rDNA is inserted into host bacterium by transformation using cold CaCl2 solution, calcium chloride. The bacterial cell containing the desired rDNA is selected using selective antibiotic in the culture medium. So basically it includes making the recipient cells competent to receive. Take up DNA present in its surroundings. The recombinant DNA bearing gene for resistance, this is of course gene of interest, to an antibiotic is transferred into E. coli cells. The host cell becomes transformed into ampicillin resistant cells. And then we move to the last and most important step in the processes, obtaining the desirable gene product. This is what our aim, ultimate aim. So the foreign DNA multiplies in plant and animal cell to produce desirable protein. Expression of foreign genes in host cells involve optimized condition to obtain recombinant protein. The recombinant cell is multiplied in a continuous culture system in which used medium is drained out from one side while fresh medium is added from the other to maintain the cells in their physiological active phase. A bioreactor provides the optimal conditions for achieving the desired product by providing optimum growth conditions, that is, the most suitable temperature, pH, substrate, salts, vitamins and oxygen. These are available in the bioreactors. So the obtaining the desirable gene product includes the cells having cloned genes of interest can be grown on a small scale in the laboratory. The cultures may be used for extracting and purifying the desired protein. After the cloning of the gene of interest, one has to maintain the optimum conditions to induce the expression of the target gene and consider producing it on large scale for business purposes. If any protein encoding gene is expressed in heterologous host, it is known as recombinant protein. And bioreactors are considered as the vessels in which raw materials are biologically converted into specific products by microbes, plant cells and animal cells, or plant enzymes and animal enzymes. It provides the optimal conditions for obtaining the desired product by providing optimum growth conditions such as temperature, pH, substrate concentration, salts, vitamins and oxygen. The most commonly used bioreactors are of stirring type, as you are seeing. The first one is the simple stirred tank bioreactor. This one, simple stirred tank bioreactor, and the second one is sparged stirred. This is the sparged stirred tank bioreactor. And here what is important is the conditions are maintained and the sterile air is also pumped. There's a motor here, foam breaker is there, flat bladed impeller is there, and of course culture broth getting rotated, and steam for sterilization, and then controlling mechanism is there in this. Similarly here, increased surface area for oxygen transfer, as you are seeing to this, gas entrainment, and then there is a movement of the rod up and down. The bubbles dramatically increase the oxygen transfer area. This is what is called sparged. And a stirred tank bioreactor is usually cylindrical, as you are seeing, or with a curved base to facilitate the mixing of the reactor content. The stirrer facilitates even mixing the oxygen availability throughout the bioreactor. Alternatively, air can be bubbled through the reactor. The bioreactor has an agitator system, as you are seeing, an oxygen delivery system and foam control system. All the three controls are there. Foam control system. a temperature control system, pH control system, and sampling ports so that small volumes of the culture can be withdrawn periodically. You can see the outlet also. So that can be withdrawn periodically. After completion of the biosynthetic stage, the product has to be subjected through a series of processes before it is ready for marketing as a finished product. The process include separation and purification, which are collectively referred to as downstream processing. So the product has to be formulated with suitable preservatives. Such formulation has to undergo through clinical trials as in the case of drugs. Strict quality control testing for each product is also required. And for all this, what is most important is the downstream processing. It involves processes that make the product obtain ready for marketing. This processes includes separation and purification. So involvement of both separation and purification is called downstream processing. Suitable preservatives are added to it and sent for clinical trial in case of drugs before releasing to market for public use. So this is how this chapter number 11 includes the principles and the processes. Of course, two principles we said, genetic engineering and biochemical engineering. Two principles are involved, and the processes we have discussed all the 6 to 7 steps, and in between we also discussed the tools that are required in making rDNA. So the whole chapter revolves around in making rDNA.